Hello, I'm Eric Archer, a former high school science teacher and currently the content strategy manager at Texas Instruments. Today I'm going to be going over the replication of DNA using the science-inspired lesson titled DNA Replication. This lesson and many more are available for free at www.scienceinspired.com for use with the TI-Nspire CX Graphing Calculator and the TI-Nspire CX Premium software. DNA replication, what does it mean and why does anyone care? DNA replication just means to make new copies of DNA. So why would we need new copies of DNA? Well, think about this. We all start off as a fertilized egg, and over time that one cell turns into trillions as we become adults. So how do we go from one tiny cell to a full-grown adult made up of trillions? DNA replication is a big part of that answer. You may recall learning about mitosis and meiosis, which are the processes that a biological system can use to make more cells. Well, during those processes, DNA must be replicated so each new cell will have a complete set of instructions. That is a complete set of DNA. So one cell becomes two, two become four, four become eight, and so on. But it's not just for growth; it's also for replacement of dead and damaged cells. So as we get older, some of the cells die. We've got to replace those. And for the why does anyone care question, well, think about this. Sometimes the mechanisms that enable DNA replication, they make mistakes, and those mistakes can turn into mutations of the DNA strand. And unfortunately, sometimes mutations can turn into diseases like cancer. So the better we understand DNA replication, the higher the probability we can actually come up with better treatments and cures for diseases such as cancer. Let's get started. Okay, so we're on page 1.1 of the activity file, and remember, you can get this activity from scienceinspired.com under the biology category, and the subcategory is genetics. It's DNA, RNA, and protein synthesis. But all of these files are free. Go ahead and download them. You can get the software from the link I mentioned earlier. And then just pull these open and start consuming them. Teachers, if you're watching this, you can edit these files, make them your own, and then send those out to the students. Students, remember, you can use these on a computer, or you can use them on a TI-Nspire CX or CX2 graphing calculator. All right, page 1.2, all living organisms carry their genetic information in DNA. As those organisms grow or reproduce, it's important that they can replicate that DNA without making errors, as we talked about before. The human genome is more than 3 billion base pairs long, so really big number. And so the idea here is think about trying to copy that much information without making a mistake. Like we mentioned earlier about making mistakes in DNA can lead to mutations, and that leads to some major problems potentially. And so it's important that it's precise. And so DNA is pretty amazing when you think about it. It's incredible that all that information is stored on just a combination of 4 nucleotides. All right, so 1.3, we're going to learn about DNA replication. We've got some simulations here that we're going to interact with and kind of illustrate the processes of DNA replication, the enzymes that are involved, some key differences from one strand of DNA to another. And so let's keep moving. Page 1.4. So why is it important to replicate DNA without errors? What might happen if there were errors in DNA replication? Well, I mean, DNA is sort of it's the blueprint of life, of your life specifically, of every organism's life. So, the bacteria, trees, fish, whatever it is, all of it contains those same 4 nucleotides in their DNA strands. And so it's important that in order for us to get the message from DNA to the rest of your body to create proteins, which are sort of foundational in all living organisms, any mistakes in DNA are going to end up being mistakes in those proteins, and that can lead to a bunch of problems, a bunch of medical problems, and unfortunately sometimes death. So replicating DNA correctly is very important. Page 1.5, when should DNA be replicated? What life events would include DNA replication? So we touched on these earlier, but growth, replacing dead or damaged cells, we need to be able to replicate DNA. to you know make the cells useful for the rest of your body. So those are some pretty big ones. And page 1.6, DNA replication is called semi-conservative. We talked about this in the DNA structure video, and this, I think this very simple simulation sort of illustrates it. So round of replication, we've got a double helix here, and when I click the little right arrow key, first round of replication, I go from 1 double helix to 2, but you'll notice the color of the ladder of each of these helices. There's a blue one blue one in this double helix and one blue one in that one. That's because we split this double helix, this original double helix, and we used each side as a template to add new nucleotides to create 2 new double helices. And obviously when I keep replicating that way, you'll see that the blue strand is from the original DNA. So this one was split, and new DNA was added to that original template. But this one was also a template, and so that's why you have a blue one here, and you've got a yellow one there. And then the same thing happened on this one. This blue strand was still used as a template to make another double helix, but so was that yellow strand. So that's why I have a yellow strand and a yellow and blue strand there. So, and if I kept doing this, I would keep doubling the amount of DNA, obviously. All right, so let's see, page 1.8. If one double helix of DNA replicates 3 times, how many double helices will there be? We saw that after 2 rounds, there are 4 double helices. So it stands to reason doubling that would make 8. What happens to the original template DNA after a round of replication? Well, I think we saw that in the simulation. The original double helix gets split in 2 single strands. Each single strand serves as a template for new DNA to be created, and as a result you get 2 double helices, each with half being the old strand and half being the new strand. All right, let's see, moving right along here, we have page 10. DNA polymerase, which is the enzyme that replicates DNA, can add about 1,000 bases per second. How many seconds would it take DNA polymerase to replicate 1 set of the human chromosome's genome? So earlier we heard that the genome is 3 billion bases long, so that's 3 with 9 zeros behind it. It's a big number, and we know that the rate here is 1,000 bases per second. So every second we're adding 1,000 bases, which is amazing. If you stop and think about that, that's incredible. So how long would it take to replicate the entire human genome? Well, let's pull open a calculator and check that out, see if we can figure it out. I'm going to use my little scratch pad, little speedy calculator icon there on the button, and we're gonna, let's see, we're gonna divide 3 billion. I got to count my zeros here, bear with me. That's 6, 7, 8, 9, and we're going to divide by the rate, which was 1,000 bases per second. And I get a number that is, let's see, 3 million seconds. Okay, so the answer is 3 million seconds. That's 6 zeros. So that's a lot of seconds. I mean, you could keep playing around with this if you wanted to. You could say, well, yeah, well, what's that divided by? And we know 60 seconds in every minute. How many minutes would that be? The answer there would be 50,000 minutes, and you can keep messing around with that and see what the hours would be and how many days and weeks and all that kind of stuff. So that's a pretty cool thing you can do. Problem 2, DNA replication is performed by many enzymes working together. Each one has a specific role that that allows replication of the long DNA molecule. Remember this, everybody. DNA must replicate from 5' to 3', and you'll keep hearing me hammer that point. That's something you need to remember. So remember it. So making replication more complex, the polymerases that add new nucleotides cannot initiate DNA. They can only add new nucleotides to a strand. So during the replication process, you have to have a starting block, what we call a primer. And in the case of DNA replication, just to make it confusing, we add an RNA primer. So there's a chunk that's added to the DNA template, We split the DNA in half, we unzip it, and each half now serves as a template. Well, in order to elongate new DNA, you have to have a starting point. That's what this is. So the primer gets added to one of the bases on the template strand of DNA, and then from there, the DNA polymerase will add nucleotides one after another. But you got to have the primer to start it. Okay. All right, so this moves us into the next section. There's another really cool simulation I'm going to go over with you in just a second. So this is just the directions page for that. Okay, page 2.3. This is the simulation, and this simulation is meant to give you a visual. It's not a simulation in that it's giving you an animation. This is just a sort of a model to show you the parts and pieces. But let's take a look. So we have a double helix, this part of the screen. You'll see, remember, DNA is anti-parallel, meaning that one half is moving in, or not moving, one half is 5' to 3', the other half is 3' to 5'. They're anti-parallel, they're opposite. So if we follow this 5' end, we should end with a 3' end, and there it is. And recall, DNA always replicates in the 5' to 3' direction, no matter what. So in order for DNA to replicate from the 5' to 3' direction, it has to have a template, and the parent DNA strand that moves in the 3' to 5' direction, opposite, because it's anti-parallel. So we make DNA in the 5' to 3' direction off of a template strand that is in the 3' to 5' direction. So what that means is when we cut DNA in half using these enzymes, we'll go over the enzymes here in a second, we're left with what's called the leading strand and the lagging strand. Okay, the leading strand is called leading because it moves in the direction of replication. So we're moving in this direction of replication, and the leading strand replication of DNA is happening from the 5' to 3' direction, which happens to be in the same direction as splitting the DNA double helix. The lagging strand, you can probably figure this out, but it's moving in this direction, and so it's a tougher strand to create because you're moving opposite the direction of the replication fork, which is what this area is called. So replication's moving in that direction. Separation of DNA is moving in this direction. Replication's happening on both parent strands, but one's moving in the direction of replication and of the replication fork, and one's moving in the opposite direction of it. So the lagging strand's going opposite the direction of the replication fork. So why is that? Again, because this strand, this parent strand of DNA, if you follow it back, 3', 5', okay? But we said that DNA always replicates, no matter what, from the 5' to 3' direction. Well, in order for that to happen, DNA's got to replicate in this way. Okay, so hopefully that makes sense. Now what I want to do is kind of show you some of the enzymes involved and what's going on with DNA. The first one you really need to know about is this one. It's called helicase. And helicase is responsible for splitting the double helix into 2 single strands of DNA, and which allows each strand to serve as being a template. Each strand serves as kind of a copy machine, where you can make a complementary DNA. So helicase unwinds that double helix and then breaks the hydrogen bonds between the bases to create these 2 single strands of DNA. So helicase, remember that one. Another one is topoisomerase. I don't know if your teacher is going to require you to understand topoisomerase or not. Some teachers do, some teachers don't. Check with your teacher to make sure. But topoisomerase is an important enzyme because it keeps the single strands of DNA from coiling back up, keeps everything kind of straight and narrow, if you will, available for copying. and so topoisomerase is an important enzyme in that regard. We also have, let's see, we'll do this one. This is polymerase 3. Now, polymerase 3 works on both the leading and lagging strand. I want to repeat that. Polymerase 3 works on both the leading and lagging strand. Even though the model I'm showing you right now only has it on the leading strand, it actually works on both. And polymerase 3 is what's responsible. It's basically the copy machine of DNA. It is what adds all the new nucleotides to the template strand to create this new double helix. And so it works, let me close this. So right now it's working off the leading strand, moving in that direction. But polymerase 3 also is what added these nucleotides going in the opposite direction of the replication fork on the lagging strand. So polymerase 3 is basically your copy machine for DNA. All right, and with that, let me show you this one. This is the polymerase 1. I know it's a little confusing, the numbers and the order of things, but it's just one of those things you'll just need to kind of power through and understand. You just really need to know what the jobs are that each of these enzymes is responsible for. But polymerase 1 also works on both the leading and the lagging strand. Even though it's only shown on the lagging strand right now, it works on both. It happens to work more often on the lagging strand because of what it does, its role. And what it does is it actually is responsible for adding DNA in replacement of the RNA primer. So remember we said that the, let's see, which one is that one? That's ligase, I think it's this one. So this is primase. Yeah, primase is kind of what starts the copying process. Primase adds a RNA primer to the complementary DNA strand. So it's the starting point. So RNA primer is added to the DNA strand, and then DNA polymerase 3 will pick up from there. It'll start from that primer, and it'll just start making a bunch of new DNA. Awesome, right? But what happens when that DNA is done and we've got to replace this primer with actual DNA, with a piece of DNA to fill that gap? We take the primer out and we got to put DNA back in. Well, that's what this guy does. This is what polymerase 1 does. It also synthesizes DNA just like polymerase 2 or 3, but it replaces that DNA. Sorry, it replaces that RNA primer when replication's just about done. Okay, so one more time, polymerase 3 is responsible for making DNA on the leading and lagging strand. It picks up from the primer, extends from the primer, elongating DNA in the leading and lagging direction. It's your DNA copy machine for the most part. All right, primase also works on both the leading and lagging strand. Primase is responsible for adding that primer. Now, because the lagging strand does DNA replication in these short little chunks of DNA, these are called Okazaki fragments. We'll learn more about those in a bit. But because it's moving in the opposite direction of the replication fork, DNA on the lagging strand has to be created in these short little chunks, which means you need more RNA primers to do that. So an RNA primer gets added, and then polymerase 3 comes along, adds a little chunk of DNA, primer gets removed, polymerase 1 comes in and fills that gap. But even with that, there's still these little areas right here, which are gaps in of themselves. There's a gap between the chunks of DNA that were added, and that's where this enzyme comes in. This is called a ligase, and ligase puts the chunks back together. It fills the gap between adjacent nucleotides. So ligase also works on the leading strand, but just not as often as it does on the lagging strand, just because on the leading strand we can make DNA, one continuous piece of DNA for a long time because we're following this replication fork. But on the lagging strand, that's not the case. We're moving away from it, so therefore we have to make these little chunks of DNA and then fill in the gaps pretty quick. So polymerase 1, primase, and ligase are active in both the lagging and the leading strand. In fact, all of these enzymes are. All of these enzymes that we're talking about today. But polymerase 1, ligase, and primase are more active on the lagging strand because they have to do these short snippets of DNA. Okay. All right. So let's check out some questions here. Which enzyme acts first in DNA replication? We talked about this in the simulation. It is the helicase enzyme. Remember, helicase is responsible for unwinding and separating the double strand into single strands, allowing each to be copied. Okay. So helicase is our answer for that one. And let's see what's next. DNA replication uses two different polymerases. Why do you think that is? So I think we've done a pretty good job of explaining the differences between them. Remember, DNA polymerase 1 is responsible for adding DNA. Also, polymerase 1 and 2 add new DNA. Both of them have that responsibility. But the difference is polymerase 3, remember this is kind of your copy machine of the cell, it's adding, it hooks onto the RNA primer and it just starts making DNA like crazy. Whereas polymerase 1, its main job is to replace the RNA primer. Once the primer's done, DNA has been elongated, we've got to fill that gap. We got to get rid of that primer and add actual DNA to it, actual nucleotide, DNA nucleotide. That's what polymerase 1 does. Does it for the leading strand and the lagging strand. Okay, works on all. 2.6, what's the difference between the leading strand and the lagging strand? We talked a lot about this already. Leading strand moves in the direction of the replication fork. Lagging strand moves away from the replication fork. The leading strand, because it moves in the direction of the replication fork, it can add long, continuous chunks of DNA. Whereas the lagging strand, that's not the case. We have to go with shorter chunks of DNA because it's moving away from the replication fork. All right, what's the purpose of primase? Some options here. Recruits other enzymes for replication. No, that's not it. Creates an RNA primer that polymerase 3 can elongate. Yep, that's definitely the one, but we'll check out these other ones just in case. Elongates the replicating DNA. Nope, that's polymerase 3 that does that. Connects DNA fragments together. You should remember that that is ligase. So our answer, what is the purpose of primase? It creates the primer, and you can't start elongating DNA without the RNA primer. You have to have it. So primase makes the primer gets added to the template strand, and polymerase 3 takes it from there. Once elongations happen, RNA primer gets taken out, and then remember polymerase 1 comes in and puts a nucleotide to replace that primer. A chunk of DNA rather. Okay, which enzyme adds new DNA nucleotides to an existing strand? All right, select all that apply. There's a hint there that it's probably more than one. So let's see, primase does not add new DNA, it adds an RNA primer. Ligase just connects the gap between nucleotides. Polymerase 1 definitely adds new DNA, and polymerase 3, that's the majority of your DNA that's added is polymerase 3. Okay. All right, now that you're familiar with the enzymes involved in DNA replication and the roles, you'll explore the differences between the leading and lagging strands. As you learn, DNA must replicate from 5' to 3' for one strand. The leading strand, this continues uninterrupted as more DNA is unwound. For the replication on the other strand, we know that that's called the lagging strand. Short segments are replicated, which are called Okazaki fragments. It's named after the scientist that discovered this process. So the lagging strand replicates with many RNA primers, which must be removed, filled in by polymerase 1, and the Okazaki fragments are joined by ligase. On the next page, you'll use a simulation of DNA to show how the Okazaki fragments were built. All right, so I was already playing with this, so I got to go back. My apologies. All right, so here we're starting fresh. So DNA is bound by hydrogen bonding. Remember that between the bases, between adenine and thymine and guanine and cytosine, they're connected through hydrogen bonding. Remember, adenine and thymine have 2 hydrogen bonds, cytosine and guanine have 3. Well, let's do this simulation, see what happens. DNA strands are separated, exposing those bases. Drag to replicate the DNA. Alright, here we go. Here's a process. So RNA primer starts DNA synthesis, which we've learned about. DNA replicates from 5' to 3'. We talked a lot about that. So on the lagging strand, DNA is going from here to there, whereas on the leading strand, it's going from here to there in the same direction as that replication fork. And so notice the bottom one, the leading strand, everything's just continuous. There's no separation, no gap. On the above one, you'll notice that the Okazaki fragments are forming, and then ligase has to come in and seal the deal there. All right, and then finally drag to replace the DNA primer and connect the fragments. Okay, well, let's see. All right, so the enzymes are coming in. DNA polymerase 1 goes in and replaces the primer, and then ligase comes in and fills the gaps. Okay, so there's a nice little, cool little simulation of DNA replication for you. All right, page 3.4. Replication of the leading and lagging strands requires different enzymes. You should know by now that's a big disagree. That is not true. Replication of the leading and lagging strands requires the same enzymes. Now, for the lagging strand, we know that some enzymes are used more often because of that Okazaki fragment issue, the lagging strand going in the opposite direction of the replication fork. So those enzymes are polymerase 1, ligase, and primase. But those enzymes I just mentioned are also used in the leading strand, just not as often. Okay. 3.5, which order best describes the order that these enzymes will act during normal replication? So let's see, we got to start. We remember what we said. We cannot replicate DNA without a primer. And which of these enzymes adds the primer? Well, it's primase. So you can't start doing anything without primase. So that rules out A, and it rules out C. We're left with B and D. Let's follow along here. Polymerase 3, yeah, that looks good. Ligase. We don't use ligase until polymerase 1 has come in and replaced that RNA primer. Remember, the RNA primer has to be taken out when elongation's done and replaced with an actual nucleotide, DNA nucleotide. And the job to do that is with polymerase 1. So ligase is at the very end. So that rules out B. We're left with D. D is correct. Primase, polymerase 3, polymerase 1, and then ligase. 3.6, how's replication of the leading and lagging strand different? I think we've touched on this quite a bit actually, but it would be useful maybe if you want just take a piece of paper out and kind of write out the steps, or write out the steps of each of the leading and lagging strands to identify those differences. So the big difference is leading strand moves in the direction of the replication fork, therefore it can be a long continuation, elongation of DNA. Lagging strand, not the case. It's moving away from the replication fork and therefore must use Okazaki fragments, which are short additions, elongations of the DNA. That requires some of the enzymes used in both to be used more often on the lagging strand. And again, those enzymes we talked about before are primase, which adds the RNA primer, polymerase 1, which takes that primer out and puts in a nucleotide, DNA nucleotide, to fill that hole, and then ligase to seal up the gaps between the different Okazaki fragments. So those are the big differences. Why are there no Okazaki fragments on the leading strand? Well, because they're not required. Okazaki fragments are only required because DNA replication on the lagging strand is moving away from that replication fork. So as the DNA gets split open, going from a hydrogen bonded bases of a double helix to 2 single strands, because DNA must replicate in the 5' to 3' direction, and in the case of the lagging strand, that means moving away from the replication fork. Short snippets of DNA must be added, whereas on the leading strand we don't need to do that because we're moving in the same direction of the replication fork. Alright, I hope this was helpful. That's the end of this particular activity. Just remember, you can download and use this activity and many more from Scienceinspired.com, and I'll put a slide up here in a second. And you can also download our software. We have some trial versions of the software that are available if you want to give it a shot. Your teacher may also have a copy of the software, or you can use these files on a TI-Nspire CX or a TI-Nspire CX2 graphing calculator. Alright, thanks for watching. Hope you found this to be useful, and please check back for more videos. Have a good day.